Hello, good day everyone. Today, we will have the continuation of our lesson on the chromosomal basis of heredity in full details. When Gregor Mendel carried out his experiments in 1860s, the biological basis of the transmission of the hereditary factors from one generation to the next was a mystery. Neither the role of the nucleus in reproduction nor the details of cell division had been discovered. Once these phenomena were understood, and when microscopy had improved enough that the chromosomes could be observed and were finally realized to be the bearers of the genes, new understanding came at a rapid pace. This lesson examines the mechanism of chromosome segregation in cell division and the relationship between DNA and chromosomes. The importance of the cell nucleus and its contents was suggested as early as the 1840s when it was noted that in dividing cells, the nucleus divided first. By the 1870s, it had become clear that nuclear division is a universal attribute of cell division. The importance of the nucleus in inheritance was reinforced by the nearly simultaneous discovery that the nuclei of two gametes fuse in the process of fertilization. The next major advance came in the 1880s with the discovery of chromosomes, which has been made visible by the light microscopy when stained with basic dyes. A few years later, chromosomes were found to segregate by an orderly process into the daughter cells formed by cell division, as well as into the gametes formed by the division of reproductive cells. Finally, three important regularities were observed about the chromosome complement of plants and animals. The nucleus of each somatic cell, a cell of the body in contrast with the germ cell or gamete, contains a fixed number of chromosomes typical of the particular species. However, the numbers vary tremendously among species and have little relationship to the complexity of the organism, as shown in Table 1. Number 2. The chromosomes in the nuclei of somatic cells are usually present in pairs. For example, the 46 chromosomes of human beings consist of 23 pairs. Similarly, the 14 chromosomes of bees, as shown in the table, it consists of 7 pairs. Cells with nuclei of this sort containing two similar sets of chromosomes are called diploid or diploid. The chromosomes are present in pairs because one chromosome of each pair derives from the maternal parent of the organism and the other from its paternal parent. Number 3, the germ cells or gametes contain only one set of chromosomes, consisting of one member of each of the pairs. The gamete nuclei are said to be haploid. The haploid gametes unite in fertilization to produce the diploid state of somatic cells. Let's take a look at the cell cycle. Mitosis is a precise process of nuclear division that ensures that each of two daughter cells receives a diploid complement of chromosomes identical with the diploid complement of the parent cell. Mitosis is usually accompanied by cytokinesis, the process in which the cell itself divides to yield two daughter cells. The essential details of mitosis are the same in all organisms, and the basic process is remarkably uniform. 1. Each chromosome is already present as a duplicated structure at the beginning of nuclear division. The duplication of each chromosome coincides with the replication of the DNA molecule contained within it. 2. Each chromosome divides longitudinally into identical halves that become separated from each other. 3. The separated chromosome halves move in opposite directions and each becomes included in one of the two daughter nuclei that are formed. In a cell that is not undergoing mitosis, the chromosomes are not visible with a light microscope. This stage of the cell cycle is called interphase. In preparation for mitosis, the genetic material or DNA in the chromosomes is replicated during a period of interphase called S. The S stands for synthesis of DNA. DNA replication is accompanied by chromosome duplication. Before and after S, there are periods called G1 and G2, respectively, in which DNA replication does not take place. The cell cycle, or the life cycle of a cell, is commonly described in terms of these three interphase periods, followed by mitosis, or the M phase. The order of events is therefore G1 or GOP1, S phase, G2 or GOP2, and M phase or the mitosis. In this representation, the M period includes cytokinesis, which is the division of the cytoplasm into two approximately equal parts, each containing one daughter nucleus. The length of time required for a complete life cycle varies with the cell type. In higher eukaryotes, the majority of cells require from 18 to 24 hours. The relative duration of the different periods in the cycle also varies considerably with the cell type. Mitosis is usually the shortest period, requiring from 1.5 hour to 2 hours. The cell cycle is an actively regulated process controlled by mechanisms that are essentially identical in all eukaryotes. The transition from GAP1 into S phase and from GAP2 into M phase are called checkpoints because the transitions are delayed unless key processes have been completed. For example, for DNA replication to be initiated at the GAP1, GAP1 or S checkpoint, some cell types require that sufficient time must have elapsed since the preceding mitosis, whereas other cell types require that the cell must have attained a particular size. Similarly, for the M phase to begin at the G2 or M checkpoint, DNA replication and repair of any DNA damage must be completed. Let's take a look at the chromosome structure. This picture shows a single chromosome, which is composed of two identical chromatids, in which one is an exact copy of the other and each contains one DNA molecule. It is sometimes called as sister chromatids. This length shows the P arm or the short arm structure, and this length shows the Q arm or the long arm structure. The centermost part of the chromosome is the centromere. This is the constricted point of the chromosome. Now let's take a look on the detailed process of mitosis. This process is conventionally divided into four stages. We have prophase, metaphase, anaphase, and telophase, or abbreviated as PMAT. The stages have the following characteristics. Prophase in interphase, the chromosomes have the form of extended filaments and cannot be seen as discrete bodies with a light microscope, except for the presence of one or more conspicuous dark bodies, each called a nucleolus. The nucleus has a diffuse, granular appearance. The beginning of prophase is marked by a condensation of chromosomes to form visibly distinct, thin threads within the nucleus. Each chromosome is already longitudinally double, consisting of two closely associated subunits called chromatids. The longitudinally bipartite nature of each chromosome is readily seen later in prophase. Each pair of chromatids is the product of the duplication of one chromosome in the S phase of interphase. The chromatids in a pair are held together at a specific region of the chromosome called the centromere. As prophase progresses, the chromosomes become shorter and thicker as a result of further coiling. At the end of prophase, the nuclei disappear and the nuclear envelope, a membrane surrounding the nucleus, abruptly disintegrates. At the beginning of metaphase, the mitotic spindle forms. The spindle is an elongated, football-shaped array of spindle fibers consisting primarily of microtubules formed by polymerization of the protein tubulin. Many other proteins and at least one RNA protein complex regulate tubulin polymerization and microtubule organization. The ends or poles of the spindle where the microtubules converge mark the locations of the centrosomes, which are the microtubule organizing centers where tubulin polymerization is initiated. So each pair of centrosomes result from the duplication of a single centrosome that takes place in interphase, followed by migration of the daughter centrosomes to opposite sides of the nuclear envelope. The spindle features three types of microtubules. One, those that anchor the centrosome to the cell membrane. Two, those that arc between the centrosomes, and three, those that become attached to the chromosomes. In spindle formation, microtubules grow out from the spindle post in essentially random directions as new tubulin subunits are added to the growing end of the polymer. For the microtubules that become attached to the chromosomes, the site of the kinetochore, which coincides with the position of the centromere. Once spindle fibers have attached, each chromosome is moved to a position near the center of the cell, where its kinetochore lies on an imaginary plane approximately equidistant from the spindle post. This imaginary plane is called the metaphase plate. Aligned on the metaphase plate, the chromosomes reach their maximum condensation and are easiest to count and examine for differences in shape and appearance. In anaphase, the proteins holding the centromeres together are degraded. The centromeres become separated, and the two sister chromatids of each chromosome move toward opposite poles of the spindle. Once the centromeres separate, each sister chromatid is regarded as a separate chromosome in its own right. Chromosome movement results in part from progressive shortening of the spindle fibers attached to the centromeres, which pulls the chromosomes in opposite directions toward the poles. At the completion of anaphase, the chromosomes lie in two groups near opposite poles of the spindle. Each group contains the same number of chromosomes that was present in the original interphase nucleus. In telophase, a nuclear envelope forms around each compact group of chromosomes. Nuclei are formed, and the spindle disappears. The chromosomes undergo a reversal of condensation until they have no longer visible entities. The two daughter nuclei slowly assume a typical interphase appearance as the cytoplasm of the cell divides into by means of a gradually deepening furrow around the periphery. Chromosome behavior during mitosis in an organism with two pairs of chromosomes at each stage. The smaller or the inner diagram represents the entire cell, and the larger diagram is an exploded view showing the chromosomes at that stage. So let us take, for example, the human chromosome is 46. If 2n is equals to 4, how many chromosomes are there in prophase? The answer is 4. How many sister chromatids are there in prophase if 2n is equals to 4? It would be 8. How many centromeres would there be in prophase? It would be 4. Well, how about during the metaphase stage? If 2n is equals to 4, how many chromosomes would there be? The number of chromosomes in metaphase is equals to 4. The same thing with the number of centromeres. We have 1, 2, 3, and 4. How many chromatids will there be in metaphase if 2n is equals to 4? There would be 1, 2, 3, 4, 5, 6, 7, 8. There will be 8 chromatids or 4 sister chromatids. How about in the anaphase stage? Take note that upon splitting of the centromere, each sister chromatid becomes a separate chromosomes. So when 2n is equals to 4, how many chromosomes would there be in anaphase? That would be 1, 2, 3, 4, 5, 6, 7, and 8. All in all, there are 8 chromosomes in anaphase if 2n is equals to 4. How many centromeres would there be in anaphase? Remember that the centromeres splitted during the anaphase stage. So there would be 8 centromeres all in anaphase. How many spindle fibers would there be in anaphase? There would be 1, 2, 3, 4, 5, 6, 7, 8. 8 spindle fibers plus the spindle fibers with unattached chromosomes. Let's move on to telophase stage. If 2n is equals to 4, how many chromosomes would there be in telophase? We have 1, 2, 3, 4. There would be 4 chromosomes in each cell during the telophase stage, or 8 chromosomes in total. How many centromeres would there be in telophase if 2n is equals to 4? There would be 1, 2, 3, 4. 4 centromeres in each cell, or 8 centromeres in total. Let us take a look on the other type of cell division, Meiosis results in gametes that differ genetically. It is a mode of cell division in which cells are created that contain only one member of each pair of chromosomes present in the premeiotic cell. When a diploid cell with two sets of the chromosomes undergoes meiosis, the result is four daughter cells, each genetically different and each containing one haploid set of chromosomes. Meiosis consists of two successive nuclear divisions. The essentials of chromosome behavior during meiosis are outlined in this figure. Prior to the first nuclear division, the members of each pair of chromosomes become closely associated along their length. The chromosomes that pair with each other are said to be homologous chromosomes. Because each member of a pair of homologous chromosomes is already replicated, it consists of a duplex of two sister chromatids joined at the centromere. The pairing of the homologous chromosomes therefore produces a four-stranded structure. In the first nuclear division, the homologous chromosomes are separated from each other, one member of each pair going to the opposite pole, and the other to the other pole. Two nuclei are formed, each containing a haploid set of duplex chromosomes, as in letter C. So we have here two cells containing a set of chromosomes. The second nuclear division loosely resembles a mitotic division, but there is no DNA replication. At metaphase, the chromosomes align on the metaphase plate, and at anaphase, the chromatids of each chromosome are separated into opposite daughter nuclei. The net effect of the two divisions in meiosis is the creation of four haploid daughter cells, in which each of the cell contains the equivalent of a single sister chromatid from each pair of homologous chromosomes. Let's take a look on the stages of meiosis, the first of which is the prophase 1. This long stage lasts several days in most higher organisms and is commonly divided into 5 substages: liptotin, zygotin, pachytin, diplotin, and diakinesis. These are descriptive terms that indicate the appearance of the chromosomes at each substage. In liptotin, which literally means thin thread, the chromosomes first become visible as long, thread-like structures. The pairs of sister chromatids can be distinguished by electron microscopy. In this initial phase of condensation of the chromosomes, numerous dense granules appear at irregular intervals along their length. These localized contractions, called chromomeres, have a characteristic number, size, and position in a given chromosome. The zygotin, or paired thread, is marked by a lateral pairing of synapses, or synapses of homologous chromosomes, beginning at the chromosome tips. As the pairing process proceeds along the length of the chromosomes, it results in a precise chromomere by chromomere association. Synapsis is accompanied by synthesis of synaptonemal complex, a protein structure that helps to hold the aligned chromosomes together. Each pair of synapse homologous chromosomes is referred to as bivalent. Condensation of the chromosomes continues. The term literally means thick thread, or pachytin. The chromosomes continue to shorten and thicken. By late packaging, it can sometimes be seen that each bivalent, that is, each set of paired chromosomes, actually consists of a tetrad of 4 chromatids, just like this one. But the two sister chromatids of each chromosomes are usually juxtaposed very lightly. The important event of genetic exchange, crossing over, takes place during packaging. But crossing over does not become apparent until the transition to diplotin. The site of exchange are indicated by the points where chromatids of different colors cross over each other. At the onset of diplotin, or double thread, the synaptonemal complex breaks down and the synapse chromosomes begin to separate, and the diplotin chromosomes become obviously double. However, the homologous chromosomes remain held together at intervals along their length by cross connections resulting from crossing over. Each cross connection, called a chiasma, is formed by a breakage and rejoining between non-sister chromatids. As shown in the chromosome and in the diagram, a chiasma results from physical exchange between chromatids of homologous chromosomes. In normal meiosis, each bivalent usually has at least one chiasma, and bivalents of long chromosomes often have 3 or more. The final period of prophase 1 is diakinesis, in which the homologous chromosomes seem to repel each other, and the segments not connected by chiasmata move apart. Diakinesis means moving apart. It is at this substage that the chromosomes attain their maximum condensation. The homologous chromosomes in a bivalent remain connected by at least one chiasma, which persists until the first meiotic anaphase. Near the end of diakinesis, the formation of a spindle is initiated as the nuclear envelope breaks down. Next to prophase 1 is the metaphase 1. The bivalents become positioned with the centromeres of the two homologous chromosomes on opposite sides of the metaphase plate. As each bivalent moves onto the metaphase plate, its centromeres are oriented at random with respect to the poles of the spindle. As shown in this figure, the bivalents formed from non-homologous pairs of chromosomes, which can be oriented on the metaphase plate in either of two ways. The orientation of the centromeres determines which member of each bivalent will subsequently move to each pole. In anaphase 1, the homologous chromosomes, each composed of two chromatids joined at an undivided centromere, separate from one another and move to opposite poles of the spindle. Chromosome separation at anaphase is a cellular basis of the segregation of alleles. Note, however, that the centromeres of the sister chromatids are stuck together tightly and behave as a single unit, owing to the presence of a protein glue that holds them together until the onset of anaphase 2, when these glue proteins are broken down. At the completion of anaphase 1, a haploid set of chromosomes consisting of one homolog from each bivalent is located near each pole of the spindle. In telophase, the spindle breaks down and, depending on the species, either a nuclear envelope briefly forms around each group of chromosomes, or the chromosomes enter the second meiotic division after only a limited uncoiling. The second meiotic division is equational, which is comparable to mitosis. It is sometimes called the equational division because the chromosome number remains the same in each cell before and after the second division. In some species, the chromosomes pass directly from telophase 1 to prophase 2 without loss of condensation. In others, there is a brief pause between the two meiotic divisions, and the chromosomes may decondense or uncoil. So chromosomes replication never takes place between the two divisions. The chromosomes present at the beginning of the second division are identical with those present at the end of the first division. After a short prophase, or short prophase 2, and the formation of second division spindles, the centromeres of the chromosomes in each nucleus become aligned on the central plane of the spindle at metaphase 2. In anaphase 2, the centromeres divide longitudinally, and the chromatids of each chromosome move to opposite poles of the spindle. Once the centromere has split at anaphase 2, each chromatid is considered a separate chromosome. Telophase 2 is marked by a transition to the interphase condition of the chromosomes in the four haploid nuclei, accompanied by division of the cytoplasm. Thus, the second meiotic division superficially resembles a mitotic division. That is all for the two types of cell division, mitosis and meiosis. I hope you learned something. See you in the next one. Bye!